Okay, this lecture is to give you some background about human papillomavirus and the paper we'll be reading. So human papillomavirus, which is abbreviated HPV, is a Baltimore class 1 double-stranded DNA, and it has a circular genome. You can see the genome here. It actually is big enough. It's about an 8 kilobase per genome, and it uses some histone proteins to keep it organized. There are about 120 some types of HPV, and we're going to talk about those types in just a little bit. Human papillomavirus encodes 8 genes. You have the E for early genes and the L for late genes. It's a naked virus, and it's about 60 nanometers in size. And the capsid shown here is made up of these two proteins, L1 and L2. So L1 is the major capsid protein, L2 is the minor. And this will become important when we talk about the vaccine for it. Like I said, there's 2 stages of gene expression. The early are all the regulatory genes, and the late are the viral capsid genes. One of the reasons HPV is interesting, and there's a lot of research on it, depending on which paper you read. Cervical cancer, 95 to 99% of all cervical cancer is caused by HPV infections. Somewhere between the 2nd and 4th most common cancer in women worldwide. You can see that HPV also does cause warts. And then there are what we call high-risk lines or types of HPV that can cause different types of cancer. So you can see lots of different types of cancer here. Again, we'll get into some more details in a minute. This is an overview of the replication cycle for HPV. So HPV enters a body through micro abrasions. It has a cell receptor. I'll show you that in a minute. It is taken in by receptor-mediated. No, let me see. Let me look at my notes before I start making these up. It's taken in, by micropinocytosis. Right? So the cell is triggered by receptor binding to take in the virus. And then the virus goes to the nucleus. So I know this is in a different language, but it's kind of cool. You can pretty much figure it out. Goes into the nucleus. So it has nuclear replication. And that's where you can see encoding happens as it enters the nucleus, replication, transcription, and of course translation happens in the, come on brain, translation happens in the cytoplasm. Sorry, I'm kind of reading notes and making sure I cover everything. Okay, so the receptor is heparin sulfate proteoglycans, which are common proteins found on, I can't write and talk at the same time, proteoglycans Also HPSG, heparin, I don't know, sulfate proteoglycan, whatever. HSPG. Oh, that's why it doesn't make sense. HSPG. Okay. Heparin sulfate proteoglycans is the receptor for binding found on epithelial cells. So human papillomavirus infects skin cells. Okay, this was just kind of translated some of that. So again, once human papillomavirus uncoats as it enters the nucleus, it actually will enter as the nucleus is breaking down. So that's why the nuclear membrane is dotted here. And this is happening during mitosis. So human papillomavirus, being a double-stranded DNA virus and encoding very few proteins, is dependent for replication on the cell's replication machinery. Right? So human papillomavirus has to infect actively replicating or dividing cells in order to get its DNA replicated as well. And it does leave the cells by lysis. And we'll talk about E6 and E7 in a minute. So here again is just a little bit about the proteins encoded, the 8 proteins encoded by human papillomavirus. We are going to be focusing on E7, but human papillomaviruses contain E6 and E7, which are important in promoting cell division, which makes sense since the cells need to divide in order to provide all the replication machinery to copy and thus make more HPV. I like this figure because it color codes the different HPV genes and kind of shows you the expression times. So basal cells, here's where the virus is going to replicate and enter. And the reason is, one, these cells have the receptors, and two, you want actively dividing cells. And what's happening in your skin cells is as you get to the top layer of your skin, right? So this is the outside of your body. You've heard that you shed lots of dead skin cells. So it's not productive for HPV to be infecting up here because the cells are not replicating. They're just being that protective layer. So you can see that the early genes, E1, E2, are expressed, and then you get E5, E6, E7, E4 expression, and then at the end you get the capsid expression. So what's happening is the HPV is in the cell, and what I want you to imagine is as the cell differentiates and moves up, gets pushed up through the layers of the skin, different HPV proteins will be expressed as the cell differentiates. Okay. HPV can also integrate. Sometimes the DNA of this virus will integrate into the host chromosome. So if you've learned about HIV, you know that HIV can also integrate. What's happening, I'm going to jump to this slide first. So what's happening is there is an important gene for human papillomavirus called E2. And E2 is important in regulating, this is the HPV genome expression. And what I want you to see is E2 is important in keeping the E6, E7 proteins at very low levels. Well, what they've found is that when HPV integrates, what they see is if this is your host chromosome, And this is your HPV chromosome. What I'm trying to show you is that in this box, the E2 gene seems to be a great integration site, but it gets split in half. Okay. So normally HPV would have its E2 gene here, and for some reason this seems to be a nice spot for breaking of the genome and integrating into the host chromosome. Why is that important? Well, if E2 is no longer regulating the expression of these genes E6, E7, then what we're going to talk about is you get an increase, so loss of E2 expression due to integration leads to an increase in E6, E7 protein levels. And that's where our issue comes for cancer. E6 binds P53. P53 is a tumor suppressor. Okay. E7 binds retinoblastoma, PRB, also a tumor suppressor. So E6 and E7 are going to affect cell cycle regulation or cell division, and it's going to become unregulated. All right, so P53 normally stops cell cycle and can trigger apoptosis. So if there are things like DNA damage, P53 gene is activated and it will stop the cell cycle. If the cell cannot fix the DNA damage, so this is showing DNA repair senescence, it can go into programmed cell death, which is called apoptosis. Okay. Apoptosis is a nice way for the body to clean up cells that have damage. What happens is when E6, okay, here's your integrated HPV genome. When E6 is expressed at high levels, it binds to P53 and leads to P53 degradation. That eventually can block apoptosis and the cell survives, even with DNA damage. HPV also has a protein E7, which binds to retinoblastoma. Okay, the way retinoblastoma normally functions is that it binds a transcription factor E27 and it stops cell cycle. So this is cell cycle genes. So RB and E27 interacting, you stop cell cycle. If everything is fine, retinoblastoma is phosphorylated, it's removed from E27, and this transcription factor can promote genes that promote cell proliferation. So when E7 binds retinoblastoma, it releases E2F, and that turns on these genes that retinoblastoma is normally regulating. So again, both P53 and RB are functioning as tumor suppressors. They're stopping cell cycle, making sure the cell is healthy and ready for cell division. Okay. If it's not, there's different pathways that these proteins can tell the cell, hang on, stop, die through apoptosis. When these HPV genes are expressed, these proteins are expressed, they interfere with this, and that leads to cell survival cell proliferation, which can lead to cancer. So as I mentioned, there's about 120 some types of HPV. They're categorized into two main types, low risk, LR, and high risk. High risk means they have the potential to cause cancer. Low risk are the ones that are associated with warts. You'll see some of these numbers come up for these types. So HPV types 6 and 11 are the most common low risk types. HPV 16 and 18 are the most common high risk types. And then there's some more types right here. And again, when I tell you a little bit about the vaccine, why don't I just jump to that, because it kind of would make sense right now. Okay, so we do have a vaccine to prevent HPV infection. It is made up of virus-like particles, which means it's empty capsid. Okay. So it's just made of the L1 proteins. You can highly express this protein in things like yeast. You put the L1s into a test tube, they self-assemble into a virus-like particle. Okay, why is that important? So we now have, as of 2018, Gardasil 9. The last type that came out in 2006 was Gardasil 4. Gardasil 9 is a mixture of these virus-like particles against HPV type 6 and 11, the two most common low risk, 16, 18, 31, 33, 45, 52, and 58. So that's why it's 9. It's against 9 different types of HPV. And if you look, these cover now about 73% of all HPV-associated cancers and the 90% of genital warts. So it's a really cool vaccine, very safe. We've talked about different vaccine types. And yes, I'll show you some other data on vaccines. Okay, so this diagram is just to kind of show you what's going to happen with low risk is you might produce warts, and produce virus that exits the cell by lysis. Okay, and this is happening, release of virus is happening here at the top of your skin. The high risk HPV will, over here, sorry, will eventually, and this is like 20 years, so it's not like it's something quick that happens. It takes a long time to transform these cells. So you're no longer shedding virus, but what you're doing is resulting in uncontrolled cell division, which is basically cancer. And the worry is that it can become invasive or metastatic cancer. Oh, sorry, I got lost in my notes. And what I wanted to say is this is all due to E6, E7 expression as shown in the red right here. So low risk, E6, E7 is kept under control. You get packaging, you get virus shedding. High risk, E6 and E7 are continuously expressed. Different types of cancer. Cervical is the most common, or the ones we worry about most. Oropharyngeal cancer, so cancer of the throat area. Oops, I had another slide. I didn't get these. Vaginal, vulval, anus, penile cancer can all be caused by human papillomavirus. How is HPV transmitted? There are alpha types that, oh, my notes are just totally mixed up here. Okay. That are primarily, that primarily infect mucosal regions. The beta are cutaneous, so at the skin level. So this is both epithelial cells, just where they're located in the body. So these are the warts, low risk type. And so the alpha primarily is through sexual transmission. So it's something to think about. This is a sexually transmitted cancer. Cervical cancer is sexually transmitted cancer via a virus. Okay, let's go. So just, I'm going to throw some stats at you because I think it's interesting. HPV is very common. 80% of people will get an HPV infection in their lifetime. But naturally, your body usually clears it. So within 2 years. So it's not, even though there's a high level of HPV out there, most of us, our bodies clear it, get rid of the virus. So 79 million Americans have HPV. More than 90% of cervical cancers are caused by HPV. And like I said, this number's in the 90s all over the place, depending on what source you look at. And the great news is it used to be a 3-dose vaccine. It's now just 2 doses. And they recommend very young people getting it. 1, you have a better immune response. And 2, they want to try to get the vaccine to boys and girls before they're sexually active, before they have this potential to be exposed to HPV. Vaccination is effective. So HPV infections have dropped 71% among teen girls, which is cool. Prevents cancer. You only have to get 2 doses, and you can get them at the same time you're getting other vaccinations. Even with all of that, the vaccination rate, this is as of 2018, is still pretty low for HPV. So people are still opting out of it. It's not one that's required for you to attend school. Some people argue that you're promoting sexual activities by telling kids they're vaccinated against a STD. I don't get it. But here's the stats. So a couple places up in the Northeast and Virginia, we've got great vaccine coverage, and a lot of other places less than 50%. Okay, so that's a little bit of background about HPV. Now what I want to do is switch to our paper. And our paper is going to focus on the E7 gene produced by HPV and its role in oncogenesis, or cancer. So, again, just to remind you, HPV E7 is going to interact with RB. RB is important for stopping cell cycle by binding to this transcription factor. RB is a tumor suppressor. It's part of cell cycle regulation. Okay. And what happens is E7 will bind to RB, releasing E2F, okay, progression of cell cycle. Here, if you want to keep track or print this or write it down, there's a whole bunch of cell lines that they're using for in vitro studies, and some are cervical, so all the white ones are from cervical cells. The ones in yellow are from different skin, so they're not cervical. Some cervical ones are positive for HPV, so they've been transformed, so they have an integrated HPV genome, so they're going to naturally express E6 and E7. The negative cell lines do not express any HPV genes because they've not been transformed with those. You might recognize HeLa. If you haven't read the book, I never get the title right, The Life of Henrietta Lacks. The Immortal Life, okay. Really good book. Let me make sure I spell her name. Yeah. Lacks. HeLa cells. Famous cell line. Taken from this woman with cervical cancer, kind of without her knowledge. Very important kind of case in what are your rights as a patient. I worked with HeLa cells for many years when I was doing my graduate work, had no idea where they came from. So if you get a chance, read this book. It's a really good read. Good story and brings up interesting ethical considerations. Okay, another protein we're going to talk about is Aurora kinase A, or Aurora A kinase. I think I always do it wrong. It's Aurora kinase A. So we've talked about kinases before. They are proteins that phosphorylate other proteins. Aurora kinase A is important in mitosis. It's located at the centrosome. So remember, the centrosome is where the spindle fibers come from. So Aurora A is very important in spindle fiber formation, assembly, stability, and therefore important for cell division. They said here, Aurora kinase A regulates spindle fiber assembly and stability, and dysregulation, so when it's not working right, is associated with aneuploidy. So the chromosomes can't separate. Aurora is this yellow, it says green, but it looks yellow, I think. And so when you have a mutation in Aurora kinase A, or you're inhibiting its function, the cells can't properly divide because the chromosomes can't properly divide. One thing that's really important that this paper does not mention is it's been shown previously that HPV E7 expression increases the levels of Aurora kinase A. Okay. Something else to put in the back of your mind is you're going to see towards the end of the paper a bunch of host proteins involved in apoptosis regulation. Aurora kinase A, being a kinase, phosphorylates a protein called BIM. Okay. When BIM is phosphorylated, we block apoptosis. Okay. There's another protein that's going to come into play, MCL1. MCL1 binds BIM and also blocks apoptosis. So normally BIM promotes apoptosis, but in the presence of Aurora kinase A or MCL1, we get a block of apoptosis. Why is that important? No apoptosis means cells can divide. Okay. Then we have this drug called alisertib. Alisertib is an Aurora kinase inhibitor. Aurora. You'd think I could spell Aurora. Okay. So, what I'm trying to show you here as you get into the readings is if you block Aurora A kinase activity, you can get delay of mitosis. They're going to talk about that. You can get apoptosis. You can get aneuploidy, where you don't have the right numbers of chromosomes, multiple nuclei, so the cell's not dividing, right? No cytokinesis, but it keeps copying its DNA. Senescence means the cell's stopping division. And sometimes you can get reentry into cell cycle. So this has a lot of cell cycle information, and I want you to remember about DNA replication in cell cycle. So let's talk about some of the techniques. So we've talked about Western blot before. The whole point of a Western blot is to detect specific protein levels. Okay. In this paper, the housekeeping gene is alpha tubulin. Sorry. A technique they're going to use is called siRNAs. This is also called RNA interference or RNAi. Okay. This is a way to knock down gene expression at the RNA level. So what's happening is you get these small interfering small interfering RNA, siRNA. And these are small, like 21 to 24 nucleotide, single strands of RNA that are going to bind your messenger RNA. And they associate with a protein complex called RISC. And RISC, once it is guided to the mRNA with the same sequence or sequence complementarity, the mRNA will get chewed up, sliced up. So if you don't have any mRNA, you don't have any protein expression. Okay, so again, you design as a researcher a small interfering RNA that's complementary to your messenger RNA through base pairing. You put it in the cell. The siRNA associates with this RISC complex, binds the target mRNA, breaks it down so there's no or reduced protein expression. So it's called a knockdown because we're not stopping RNA transcription. So we're not knocking out gene expression. And what you'll see in the paper is you don't get 100% knockdown. So let me show you that. So Western blot plus siRNA, what the data looks like. So you're going to see some data in these figures where they've used an siRNA to knock down protein expression. So a control siRNA in our paper is called NT, non-targeting. So just a random sequence of nucleotides that's not going to interfere with anything. And you shouldn't see any difference in protein expression. So they're looking at the wave protein. Okay. When you put in the siRNA that's specific to wave 1, you see that there's less. So remember, the darkness of a band tells you kind of quantity. So less dark band means less protein is being expressed. So Western blot, again, is for proteins. And you'll see that different siRNAs are better or worse at knocking down expression. Wave 2, here's the result of the siRNA, and WASP, here's the result. In this case, GAPDH is their housekeeping gene, so loading control to make sure everything's fair. And then you can also take images and you can get the pixel numbers of these different bands and you can plot them. So you can see how much the wave siRNAs here are decreasing protein expression. In these figures, they're doing drug sensitivity, and the way they're doing this with this resazurin assay is that they're looking at the number of live kind of to dead cells, right? So the more live cells you have, the less toxic something is. Okay, so pink is live or viable, purple is dead, right? And you can quantitate this color to give you, you can plot it and give you a value. And so what they do is they calculate IC50. We talked about this before. Concentration of an inhibitor where the response is reduced by half. So you get a value, and what I want you to remember is that the smaller the IC50 value, the more sensitive the cells are to the drug. Okay, so what they're going to do is they're going to look at that alisertib drug and see what its effect on cell proliferation, cell death is. And if it takes less and less of that drug, right, over here on your concentration, it's going to have a smaller IC50 value. Okay, after a lot of in vitro stuff is done, we move to in vivo. 2 sets of experiments. The first one is that they're going to inject tumors. This is supposed to be a little, let's see, subcutaneously. So right under the skin, they're going to inject some of those cells that they've been working with, right? So like HeLa cells. And because those are cancer cells, the mice will eventually develop a tumor in that area. And so what they're going to do is they're going to measure the size of this tumor after they add this drug and see if alisertib is affecting tumor formation from HPV infected cells. The second type of in vivo experiment they're doing is on K14. I just lost. Sorry. Oh, sorry. I should have known. K14 E7, which means these are transgenic mice. And they have human papillomavirus E7 protein in all their cells. But it's under a keratin promoter. So it's only expressed in the cell, in the skin tissue. So then what they want to do is they want to see what this drug does. And so they actually remove a patch of skin from these transgenic mice. Right? And they graft them on to genetically similar mice. Okay. This is the only place where E7 is expressed, in these skin grafts. Right? And then they're going to give these new hosts with the skin grafts, alisertib to see the effect on these skin grafts, these E7 expressing cancer cells. And then they're also going to say, well, what's the effect on normal cells? Okay. So you can't give it to these mice because they express E7 everywhere. I mean, not everywhere, but in all their skin. Sorry. These guys now just have a patch of E7 expressing. So you can see, like if you had skin cancer, right, it would not be throughout your across all your skin. It would be in an area. So now you've got normal cells to look at and the cancer cells and looking at the effect of this drug. Flow cytometry, we've talked about this. This is a way they're using to determine the amount of DNA in a cell. Okay, so they can use stains that it's a DNA binding stain. And remember that as you go through cell cycle, your amount of DNA changes, right? So you're a 2N diploid organism. In S phase, you're 4N. I don't know why I put greater than. Right? You've duplicated your chromosomes. And then after you undergo mitosis, you now have 2N cells again. You can use flow cytometry to figure out how much DNA content there is based on how much dye, and then they can correlate that to what phases of the cell cycle. So normally most cells are in a G1 phase, right? Some in S2, and then G2, they can tell by the way the DNA content changes. So they're going to use flow cytometry in figure 4 to try to figure out what's going on in these cells based on how much DNA is there. For figure 6, they're using, I'll just make sure I did this right. Yes. Figure 6, they're using a GFP reporter construct. Okay, so we've talked about GFP, green fluorescent protein. And what's happening here, I just want you to see, is that they've made this virus, it's a recombinant virus, to infect cells, and it has GFP, and it has E7, okay? And so whenever you see green, it's telling you that there's E7 also being expressed. This is a way, the value of this, I hate this. Look at live cells and approximate E7 levels. Okay, so if we did a Western blot, we'd have to kill the cells, grind them up, run the SDS page, and blot them. We can do FACS, which is cell sorting based on flow cytometry, and you can put, for instance, the high and the low, that's how they spell it, GFP expressing cells, which will mean you have a population you can sort into test tubes of high expressing E7 and low expressing E7, because the E7 levels are approximate to the green fluorescent protein levels. So what they're doing here is they're looking, does it matter how much E7 is being expressed as to how alisertib works. Another kind of assay they're doing for figure 7b is time-lapse microscopy. And so what they can do is they can take snapshots of the chromosomes and the cells dividing and figure out how long they're spending in mitosis. So I'm going to show you this video real quick. And it goes really quick. 9 seconds. Watch the cell divide. Okay? That was 7 hours and 35 minutes in 9 seconds. Show you one more time. So they can take images, put it all together, figure out the time that it takes a cell to divide. And you're going to see with drug treatment or levels of E7, you're going to have different times in mitosis. Finally, but it's actually really figure 6, brings in all these proteins, anti-apoptotic proteins, promoters of apoptosis, signals of apoptosis. So keep this little table handy as you look at figure 6. I think that's figure 6 and figure 5. Sorry. Okay, so they're trying to figure out what's happening with E7 and Aurora kinase A and this drug. So again, I'm going to post this on the front home page. This will help you kind of put some of these into perspective as to what's going on in this paper. Important to look at these genes of interest, and then that, where is it? Cell line, keeping track of what all these different cell lines mean. All right, I hope that gets you going on the HPV E7 paper. Thanks. Oops.